Welcome to our cell biology course, and let's start right now with chapter number one, a preview of cell biology. We're going to talk about six main topics in this class. In the beginning, we're going to cover a brief history of the cell theory. So we're going to talk about microscopy and what are the progress in microscopy that needed for the development of the cell theory. And we're going to talk about what is the cell theory. Then we're going to move into the different cellular dimensions, yeah, how big the cells are from the smallest one to the largest one. And then we will move into how different fields work together, the development in different fields allow to development of cell biology. And we're going to talk about biochemistry, we're going to talk about cytology, and then we're going to talk about genetics, how these three main fields work together to develop this area of cell biology. And at the end, we will finish talking about a little bit about the scientific method, in particular in terms of cell biology. So let's start. So let's start talking about history, the historical approach of the cell theory. Here you can see some examples of different questions that they could be in the quiz, or they could be in the test. So questions related to the emergence of cell biology and which fields are important to that. In addition to that, a second question related to Robert Hooke. Yeah, so the history of cell biology. The third question here related to what are the characteristics of a model organism? That is pretty easy that you have covered maybe in previous courses, and we're going to review it, seeing it from a cell biology perspective. And finally, very easy questions like which organelles store the DNA in plants and animals. So this is the first class, so these will be easy questions. So in addition to those easy questions that I'm sure that you will be able to answer, there could be questions that they will start getting more difficult. So this one is one of those ones. So here, the question is, which of the following is closest to a micrometer in size? And here you will see different structures in the cell, like the DNA strand, the ribosome, the length of a plant cell, a typical prokaryotic cell, the length of a chicken cell. So uh, you can start guessing right now, but during the class, I will cover both the easy questions and the difficult questions, and hopefully you will be able to answer all of them, and you will do very well in the quiz and in the test. So this slide summarizes all this lecture. I want that you go home and you remember that the cell is the basic unit of life. So why we study cell biology? Because we need to study the form, the structure, and function of cells. And I will talk a lot about all these functions and structure and forms during the entire semester. And today I'm just going to give you a little bit of ideas that you need to remember. And why cell biology is so important? Because in addition to understand all these form, structure, and functions, we need to compare all those cells, and we can study what their interplay can tell us about different living organisms. And there are so if you remember that the cell is the basic unit of life, you start asking questions. Okay, so what appeared before? The DNA or RNA or the proteins. So if we know that this is the unit of life, how the first cells appear and how multicellularity appear, why cells are constantly changing in our organism. So in addition to have multicellular organisms to have different tissues, so a neuron is different from an epithelial cell, from uh, there are so many different type of cells, but all of them they divide. Yeah. So what are the implications of cell division? So for example, what are why cancer appear? And we will give some examples that are very interesting, and this is why we study cell biology. And key how cell biology appear. So I will talk about these three main fields. So one, it will be genetics. Second, biochemistry, that some of you have taken biochemistry, and one that maybe you haven't here, that is cytology. Cytology cytocell logos study. So cytology will talk about the difference between cell biology and cytology in the future. But the convergence of these three different fields, at least, is three disciplines were the one that create cell biology. And if you remember from previous semester, genetics start around more than 100, less than 200 years ago with Mendel. So cell biology is even newer, and so it's a new and exciting field in biology. In your textbook, this chapter is divided in three main sections. In the beginning, they talk about the historical approach to the cell theory, and I'm going to start with that. Then I will move to the emergence of the modern cell biology, and finally, I will talk about about the scientific method, and I'm going to in particular focus on the scientific method in terms of cell biology. So let's start. So let's talk about the history of the cell theory, and we're going to start talking about two main scientists. So these are the two scientists that we're going to talk about. One that is Robert Hooke, and the other one that is Anton van Leeuwenhoek. They were responsible for the early discoveries in microscopy. And if you see, both were European men. One lived in England and the other one in Netherlands, and they live around the same time. So let's talk about first about Robert Hooke. So the first thing that I want that you remember is that this guy, Robert Hooke, is one of the earliest scientists to use a simple microscope. And here you can see a picture of Robert Hooke, and here you can see a picture of the first lenses that he creates, yeah, these magnifying glasses that were used as a microscope. And second thing that I want that you remember is that he didn't observe living cells, yeah. What he observed was dead cells, yeah. So, and he observed it in a cork, and he used the microscope, and he found that those compartments that he saw, they looked like the cells in a monastery, and this is why they are called cells. He gave them the name, and of course these cork are mainly, what he observed was the cell walls of a dead plant tissue. So again, he didn't observe living cells. He observed dead cells, and he was the one who gave the name of cells to this type of compartments. So for the quiz, there is a lot of information about Robert Hooke, but I want that you remember just three things. First thing, that he didn't observe living organisms. He studied dead plant cells that they were finding a slice of cork. The second thing that I want that you remember is that this word in Latin, cella, yeah, that means little room, and the cellum is the one that in English is cell. In Spanish it will be actually célula. But anyway, and the third one, that is that he used a microscope that it was 30 times of magnification. Okay, these three key points that I want you to remember about Robert Hooke. Okay, so now let's try to answer this question together. So you can stop the video, you can read it with me. So here, Robert Hooke coined the term cell. We just studied that when studying thin slices of a cork. Perfect. This, okay, we have to fill here this blank, were the first cells observed because, and something else. Okay, so here you have 5 options. So I want that you read them, stop the video, and then I can help you to find the answer. Okay, so I'm sure that you stop the video, or if you don't want it, I will tell you that the first thing that you need to do is to discard the question. So here they are talking about that those cells, some characteristics of those cells. So here you see that those cells were dead plant cells. So this sounds like good. like those one were dead. The core has dead plant cells. And here they said immune cells, so this one is already wrong, so B is wrong. These little rooms, you know, this is not this is not perfect. Plant cells will be better, so we need to go and look the rest. Dead animal cells, this is wrong already. And these compartments, this is not perfect. So far, A is better. And okay, so those dead plant cells or little room or compartments were the first cells observed because the thick cells wall didn't require high resolution of magnification to view. That makes sense because if you remember, it was only 30x. Yeah, so that sounds good. B is already wrong because here they were 100 nanometers in diameter, much larger than most plant cells. This doesn't make any sense. And yeah, nanometers is very small, so that is wrong. They were actually the result of multiple cells that had merged and died to form that. That is not true. So um, here the best answer will be A. So let me show you in the next slide. Okay, so the answer pretty easy. The only one that work is dead plant cells. C and D little rooms partially work, but the next part, this size is wrong. And what they are saying about fusion of cells, this is this is too complicated and is not related to the topic that we are talking. So A is the best answer. So pretty easy. Hopefully you got it right. Okay, so we talked about already Robert Hooke. Now we're going to talk about the other scientist. And if you remember, the other scientist was Anton van Leeuwenhoek. This very pretty piece of metal that is of the size of possibly one of your fingers. It was the first microscope. And the most interesting is that this was 300 magnification. Yeah, so the compound microscope that we have seen in the generally you have seen in the labs in high school or here, this is 3 times more powerful. So it's fascinating how with these simple lenses he were able to get this magnification. And different from Leeuwenhoek that he observed that cell, he observed actually live cells like blood cells, sperm cells, bacteria, and unicellular organisms that he found in pond water. So this is fascinating, and as you see, those two scientists were observing different things and they start documenting and reporting all the things that they were observed. This magical word that it was microscopic. So now that we talk about these guys, we're going to talk about the importance of microscopy that allow detailed studies of cells. And during the semester, we will talk about multiple type of microscopy and how those were very important. Without them, there was a restriction in the progress of cell biology in the beginning. If you remember, when we talk about genetic revolution, as there was not a lot of resolution, yeah, there was limited resolution and power in the magnification. So if you remember the homunculus, that humans were inside of the sperm and inside there was another tiny human. That was just because we didn't have good microscopes. So that was a limitation. And the other limitation is that we need to start thinking differently. So not only describing cells was important, but we need to start emphasizing on mechanism, on the explanation. For example, not only that cells are moving, but studying, for example, the proteins that are involved in the movement. So what happens if we remove those proteins? The cells stop moving. Maybe they play a role in that. So during the semester, we're going to cover different type of microscopies. Some of them you will see it in the lab. Some of them you will use those microscopes. In our cases, we are just going to briefly talk about that during the class. And when we read papers and during the class, we will talk about some observations and some mechanisms. Yeah, so how the cells start working, what are the proteins, and how they found those things. So to finish this section, we're going to talk about the first type of microscopes that we still use, that are the compound microscopes. And the compound microscopes are one of these one. There are different types, but by 1830 they become uh, common everywhere, and they have two lenses that allow the improvement of resolution and magnification, and they were able to observe 1 micrometer, even structures of 1 micrometer. Okay, so that was a pretty good advancement. Yeah, so from these ones to the compound microscope that we still use, and we are going to cover now many more improvements that took place. Okay, with this, we finished the historical approach of cell biology, and now we're going to move into the cell theory. Okay,